So you know what's interesting? Over 99% of the estimated 1 trillion microbial species have not yet been identified. Taking a quick peek at the phylogenetic tree of life reveals the enormous diversity among unicellular organisms. Given the known diversity in multicellular organisms, a tiny portion of the phylogenetic tree over here, I'm sure you can imagine just how interesting and diverse the world of microorganisms is. And so, keeping in mind this diversity, today I would like to discuss a specific way microbiologists are utilizing microbial diversity in biotechnology, and that's bioremediation. First, we'll cover how microbial metabolism relates to bioremediation, and then we'll take an in-depth look at the reduction of uranium-6 by metal-reducing bacteria. Okay, so first things first, bioremediation is the use of naturally occurring or genetically modified microorganisms to break down, sequester, or transform environmental pollutants. Microorganisms are ideally suited to the task of contaminant destruction because they possess enzymes that allow them to use environmental contaminants as food, and because they are so small that they are able to contact contaminants easily. I'm sure you're all quite familiar with how human beings acquire energy in aerobic respiration. Our cells strip electrons away from glucose, shuttle them along an electron transport chain, and drop them off on a molecule of oxygen. Metabolic regimes can be summed up on scary-looking charts like this one, but for our purposes, all we really need to focus on is this chemical equation. The main thing you need to know to understand bioremediation is that organisms need an electron donor and an electron acceptor to fulfill their metabolic needs, indicated here by the arrows. Now, what makes microbes so cool is that they are able to use an extremely diverse range of substrates as electron donors and acceptors. The coupling of electron donors and acceptors during redox reactions, shown here, is essential for the series of electron transfer reactions that drives metabolism through the electron transport chain. Here's a chart depicting important redox couples, many of which are utilized by microorganisms to fulfill their metabolic needs. Some microbes even contain the genetic arsenal necessary to carry out multiple of these redox reactions, depending on what is available in their environments. So you can imagine the incredible diversity of metabolism that exists in the microbe world. And this is going off of what has been observed in less than 1% of the estimated total microbial diversity. Now, as can be seen in various biochemical mechanisms, for instance, the binding of both oxygen and carbon monoxide to hemoglobin, interactions between enzymes and substrates are usually very specific, but can exhibit some variation with similar molecules. And this is the basis for most microbial bioremediation. Take the iron-3 reducing bacteria species Geobacter metalloreducens, for example. These guys grow under anaerobic conditions by enzymatically coupling the oxidation of acetate with a reduction of iron-3, as described by this chemical reaction. Back in 1991, microbiologist Derek Lovely and his team had an interesting hunch. Based on thermodynamic calculations of the energy transferred per electron, the reduction of uranium-6 to a uranium-4 precipitate has the potential to release more than twice the energy per electron available from iron-3 reduction. Therefore, if our iron-reducing bacteria could use toxic uranium-6 as an electron sink instead of iron, and transform it into non-mobile electron-4, it would halt the spread of uranium contamination in groundwater and support energy production for the bacteria. So they tried it out, and that's exactly what happened. The Lovely Lab grew G metalloreducens anaerobically on media that contained iron-3 and acetate as the sole metabolic substrates, and then they transferred the culture over to media lacking iron, that instead contained uranium-6 as the potential electron acceptor. Remarkably, the concentration of uranium-6 decreased over time, indicated by the squares on this figure, and uranium-4 increased over time, indicated by triangles. Indeed, cell growth coincided with uranium reduction and stopped as soon as uranium-6 became depleted. In another experiment, when carbon-14 labeled acetate was used as the electron source, carbon-14 labeled carbon dioxide was generated in direct proportion to uranium-6 reduction. Microbial involvement in uranium reduction had previously been considered limited to indirect effects, such as microbes providing the necessary substrates for abiological redox reactions. However, these results indicated that in fact G metalloreducens was using acetate to reduce the oxidized form of uranium to support their metabolic needs, according to this equation. Now, the bioremediation field has developed a lot since 1991, and we've identified bacteria that are able to transform all kinds of chemical species. Many are able to do so much more rapidly than abiological reactions. However, there are plenty of complications. For one, it's no simple task to optimize the growth conditions of a given bacterial species in situ, and not to mention concerns about introducing non-native, possibly invasive microbe species into new environments. However, don't lose hope, for it's clear that the bioremediation field is beginning to pick up speed, and I personally think that the key to developing this field is unlocking more microbial diversity and further exploring the chemistry of life. Thanks for watching!